Hello, welcome back for more biotechnology. So, now that we have a pretty good idea of what DNA synthesis is all about, especially in vitro DNA synthesis, let's talk about what we can actually do with some of those in vitro DNA synthesis products. So, we already covered in a previous chapter, I think it was the last chapter, that one very common thing that is done is that DNA is synthesized artificially, right? So we're not using cells to make it. We can actually use machines to actually take samples of our oligonucleotides and put them together, and we can actually synthesize short little segments of DNA called oligonucleotides. So we've already seen that we can use these oligos for applications like southern blotting. So that would involve making an oligo that is labeled with some sort of fluorescent probe, and then once we get that probe to bind to a targeted DNA sequence based on complementary base pairing, we can detect with our fluorescent probe whether that actual sequence is present or not. So a good example of what we could use something like this for is that because we know that the pancreas is the organ that produces the hormone insulin, which we've talked about extensively throughout the semester, if we get extracts out of the pancreas, out of the pancreatic cells, we should be able to find a high amount of insulin messenger RNA. So if we design a labeled fluorescent oligonucleotide that is complementary to that messenger RNA, we should be able to detect that particular message within the cells of the pancreas versus, say, the cells of the brain or the cells of the skin, the cells that don't actually make insulin. So that can be very, very useful for us in determining which types of cells or tissue express a particular type of message versus the cells that do not. Okay, so we've talked a little bit about the southern blot before, but now we can actually talk about it in a little bit of detail. The southern blot, the whole purpose of it is to detect DNA molecules after they have already been run through gel electrophoresis. Whether it's an agarose gel or an acrylamide gel, it's just going to depend. But the whole process is pretty much the same as what we saw with Western blotting. So once you have run out the gel is going to be overlaid with a positively charged membrane, and you'll hopefully recall that all DNA molecules are negatively charged, so there should be a natural affinity between the DNA molecules and this positively charged membrane. So we can transfer the DNA molecules to the membrane and then lock them into place with UV radiation. You can see the UV crosslinker down there in the diagram. And then we can incubate the blot that has our DNA molecules bound to the membrane. We can incubate this all with our fluorescent probe. Our fluorescent probe should bind to any DNA targets that are complementary, and then we can visualize the blot by shining a particular wavelength of radiation on them, hopefully the right wavelength to get our fluorescent probe to fluoresce. We can do something very similar to detect RNA molecules specifically, called a northern blot. And then we've already talked extensively about Western blotting and how this is used to detect particular proteins. But as we've said before, because proteins do not bind each other with complementarity in the same way that nucleic acids do, we had to use antibodies instead of fluorescently labeled oligonucleotides. Another thing that we can use oligonucleotide probes for, as we touched on in a previous chapter, is high throughput assays involving the use of what are called microarrays, or little gene chips that contain thousands of microscopic wells that we can incubate different samples and probe for different DNA sequences with our fluorescent probes. The most common of these is made by a biotechnology company called Affymetrix. The idea, as I said, is that thousands of DNA samples can be loaded into the tiny, teeny, tiny wells on the chip, and then we can overlay them with the probe. If we have a chip reader, we can actually expose each of those wells to the wavelength of light that our fluorescent probe is specific for, and we can generate a digital heat map that indicates the degree to which our probe annealed to a particular DNA sample. In other words, how much of the target DNA was actually there. The general idea is that the red and yellow areas indicate a large amount of that DNA that was detected, and the blue regions indicate a very low amount of DNA or none at all. So if we know exactly which samples were loaded into which wells, we can easily pick up which ones are lighting up hot for that particular DNA sequence and which ones are cold. That's why it's called a heat map, hot and cold. Okay, so the last thing that we're going to talk about in this video, and it is going to be a little bit of a discussion here, is primer design. One of the most important technologies, one of the most important reasons why we synthesize oligonucleotides is for a technique called polymerase chain reaction, or PCR. The actual PCR technique will be discussed in the next video in this series, but we want to go ahead and talk about the particulars of synthesizing the primers that are necessary for PCR and also DNA sequencing. So actually constructing these primers is going to be very important, and we have to be very careful about how we do it for a couple of reasons. So not only does our primer have to be able to anneal to the DNA that it's going to bind to, but we also have to have it fall off when necessary. So for that to be possible, the idea is that we need to design a primer that is going to bind to the template tight enough for it to stick, but we don't want it to be too sticky. We don't want it to bind too tightly or too loosely. We can run into problems if either one of those occurs. So the general guidelines for designing primers, usually for PCR, are this. So the primer should contain a sequence that is complementary to somewhere between 20 and 30 base pairs of DNA in the template. In my experience, I usually find that about 21 is usually the best. It doesn't have to be exactly 21. You usually like it to fall somewhere in that 20 to 30 area. The next guideline is that the GC content, the guanine cytosine content of the primer, should fall somewhere between 35 and 65%. Now, this guideline gets into what we were saying before about not wanting the primer to bind too tightly or too loosely. You'll hopefully remember that guanine cytosine base pairs are stronger than adenine thymine base pairs because they have an extra hydrogen bond. So they're about 1.5 times stronger than an AT base pair. So for that reason, if your primer contains too many guanines and cytosines, that is going to make the primer bind to the template more tightly. So if you have too many of those, you're not going to be able to get the primer to come off of the template. But if you have too few of them, your primer is going to struggle to bind to the template at all. So for that reason, we want to limit the GC content to be somewhere in this range here, which usually isn't too difficult. And then finally, another major thing we want to avoid is we want to avoid having repeats of the same nucleotide over and over again. You don't want to have a primer that goes A, T, 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 because what can happen is that creates more opportunity for the primers to bind to each other, which is what we call primer dimers. If you get a primer dimer, those primers are going to be totally non-functional, and then at that point you're going to be wondering what went wrong. So you want to try to have primers that don't repeat the same nucleotide more than maybe twice at a time. So what PCR is going to involve are extreme manipulations in temperature in order to make the technique work, and we'll talk about that in the next section and why that is necessary. But because the temperature of the PCR reaction is going to be varying quite a lot, we need to take into consideration the melting temperature of the primer itself. Now, when you see the word melting, you're probably thinking of butter melting into a liquid. You're thinking of a solid turning into a liquid, right? Well, that's not exactly what we mean here. The melting temperature is the temperature at which the primer is going to fall off of the template DNA. So whatever the melting temperature of the primer is, this basically tells us at this point the primer is basically 50-50 either bound to the template or it's fallen off. So what we would like, and you'll see why in the next section, we would like our primer to have a melting temperature of about 60 to 65 degrees Celsius. The idea there is that when the temperature is much lower than 60 to 65 degrees, then the primer should comfortably bind to the template. But if the temperature is above the melting temperature, then the primer is going to fall off. So the temperature manipulations that happen during the PCR reaction are going to involve both well below and well above the melting temperature. We need those temperatures to happen at the right times. So thankfully, there are web tools available online. You can find a number of these. This one I just so happen to find is the one I'm using as an example here, where you can input your proposed primer sequences, and the program will actually predict the melting temperature, as well as tell you what percentage GC content you've got. So in this particular example, you'll notice that I have two different primers listed here. The first primer goes ATC, TAC, TA, GGC, TAAC, TAGC, CGC. You'll notice that no particular nucleotide is repeated more than twice at a time, which is good. You'll notice the same thing for the second primer. And then you'll see here that we get the given parameters. Both of them are 21 nucleotides, which that's great. That's exactly what we want. The first primer has 48% GC content, that's fine. The second primer has 52% GC content, that's fine as well. And then they each have a melting point of either 63 or 64 degrees Celsius. So this is perfect. So we can move ahead with ordering these primers from a company that synthesizes them, like Integrated DNA Technologies, or IDT. So hopefully the case will be, if you're designing primers, that everything is perfect and you don't have to make any adjustment. But that's not always the case. If you propose a primer and it does not fit the given parameters, basically what you're going to have to do is that you either have to shorten or lengthen the complementary sequence until you get the parameters that fit. So we'll look at a particular example here in just a minute where we actually have to do something like that. So, as I was saying, let's get a little bit of practice with this so we can see what we have to do when our primers do not meet the given criteria. So let's say that we want to design primers to amplify the insulin gene so that we can insert it into a vector. Okay, so first thing that we need is we need the entire DNA sequence of the insulin gene with any non-coding gene sequences, which are called introns, taken out. So for this, we are going to get what is called the cDNA sequence, or copied DNA sequence. cDNA is generated from isolated messenger RNA after the introns have already been removed, and then messenger RNA is exposed to an enzyme called reverse transcriptase, which synthesizes a DNA copy of the messenger RNA, which is why it's called a copy DNA sequence. And then we can actually get this sequence deposited into online databases found on NCBI. So what you're looking at here is the cDNA sequence for the messenger RNA for insulin. So you can see a whole bunch of nucleotides here. You'll notice one thing that you'll probably notice right off the bat is that there are a whole lot of A's at the end, at the 3' end. That's normal. That's normal for a messenger RNA. Messenger RNAs have what is called a poly-A tail, which helps the messenger RNA to be recognized by the translation machinery. But we don't really need it for making PCR primers, so we're going to end up ignoring it, so don't worry about it too much. Okay, so now that we have the cDNA sequence, we want the start codon and the stop codon, which gives us the open reading frame for the insulin gene. So the first thing we're going to do is we're going to start at that first nucleotide, and we are going to scan from 5' to 3' until we find our start codon, which is always ATG. So you can see it takes us a little while to find it, but we do eventually get ATG, which of course codes for the amino acid methionine. So if you look at the translation up here, you'll see that the first amino acid in the sequence is indeed methionine. Okay, so our stop codon doesn't happen until about right here, TAG. So that's the DNA sequence. In RNA terms, the stop codon used here is UAG, which again is one of our 3 stop codons. So this is going to mark the end of the open reading frame. So from the methionine that is read at ATG, Every 3 nucleotides after that, GCC codes for alanine, CTG codes for leucine, TGG codes for tryptophan, and so on. We are going to read in frame 3 nucleotides at a time until we hit that stop codon, and each codon is going to give us a particular amino acid in the insulin protein sequence. So like I said, you'll remember that the start codon, which is AUG and mRNA, sets the frame for how codons are read, so we really want to make sure that when we're designing our PCR primers, we want to make sure that we amplify the genes so that we start at the start codon and we end at the stop codon. We don't really want or need any of the stuff that happens before or any of the stuff that happens after. We just need the part of the gene that codes for the protein. Okay, so since by now we know kind of what parameters we're after in designing our primers, we want each of our primers to have complementary sequences between 20 and 30 base pairs. 21 is usually a pretty good starting point. So let's start with getting the first 21 base pairs of the insulin reading frame like what you see here. We get the start codon there in green, and then we get the next 18 nucleotides after that. Okay, so if we plug that into our web tool that is going to give us the GC content and the melting temperature, you'll see, okay, we've got 21 nucleotides, 67% GC content, which is a little on the high side. We could probably live with that if we had to, but that's not really the main problem. The main problem is the melting temperature. The melting temperature is way, way too high. We want it to be between 60 and 65 degrees Celsius. It's at 76. So that's kind of a big problem, right? So it's not really ideal because at 21 nucleotides, we're already on the low end of that 20 to 30 base pair guideline. It's not ideal, but we're going to have to end up deleting some bases off of the 3' end so that we can lower the melting temperature and get that GC content down. So working with it myself, I had to delete 5 base pairs off of the 3' end just to get our melting temperature and our GC content down to something that we can work with. 66 degrees Celsius is still a little too high, but if we shorten the primer any more, we're really going to run into problems. So we're just going to have to try to live with this as our forward primer, even though it's shorter than we would like and the melting temperature is a bit higher than we would like. But there's not really anything that we can do about the first 20 or so nucleotides in the insulin gene. So that's going to dictate what we have to do here. So this is probably the best that we can do at this point. Okay, so now that we have our forward primer, we need a primer that will bind to the end of the reading frame and catch the last few codons, including the stop codon. So if we copy the first 21 base pairs of the end of the reading frame, we get CTG, GAG, AAC, TAC, TGC, AAC, and then finally the stop codon TAG. But here's the problem. We can't just roll with this as our primer. You'll recall that DNA is always written from 5' to 3' in the same way that DNA is always synthesized from 5' to 3'. So if we make a primer that corresponds to what we have here, starting with CTG, all it's going to do is it's going to bind to the other end of the same strand that is bound to the forward primer. And that's not going to do us any good because if the forward primer binds the way it's supposed to, we're going to handle the end of that anyway. So we want our reverse primer to bind to the complementary strand to what the forward primer bound to. So this is not actually the sequence that we're going to use for our reverse primer. What we're going to need to do here is get the reverse complement sequence and write that from 5' to 3'. So currently, as you see it here, this is written from 5' to 3'. So if we get the complement for it, we would get G, A, C, C, T, C, T, T, G, A, T, G, and so on, but that itself would be written from 3' to 5' as I just verbalized it, so we would need to reverse that. So if we do get the reverse complement, we get this right here, CTA, which is complementary from right to left, to the GAT. So our reverse complement sequence would be this right here, and this is going to be what we then plug into our web tool. So you'll notice here we have 21 nucleotides, we have 48% GC content, and a melting temperature of 62 degrees. This is just fine. We don't need to make any further alterations, so this is going to be what we call our reverse primer. So to conclude, if we're going to amplify something like the insulin gene by PCR, the primers that we would need synthesized for us by a company like IDT would be the forward primer, 5' to 3' would be ATG, GCC, CTG, TGG, ATG, C. And then the reverse primer from 5' to 3' would be CTA, GTT, and so on. So ordering these primers would be very simple. You would just go to IDT's website, paste these sequences in, they'll get the order, they will synthesize these primers for you, and then they will send them back to you. It's easy peasy. The design of the primers is what is actually difficult. So in addition to this, we can also engineer restriction sites for enzymes like BAMH1, which we've talked about before, which is GGA-TCC. We would just stick that onto both the 5' end of the forward primer and the 3' end of the reverse primer if we want to stick our PCR product into a plasmid vector. And we don't have to worry about these restriction sequences affecting the melting temperature because they are not currently complementary to anything in the template DNA, so it's not going to affect how the primer actually binds to the template. All right, so that is going to do it for our discussion on primer design and other sorts of things that we can do with synthesized oligonucleotides. So join us next time and we will have our next discussion after primer design. We will actually start talking about what we use those primers for in the process of doing polymerase chain reaction. So thanks for your attention, and I will see you next time. So long.